We completed cDNA amplification. Now we will perform SPRIselect cleanup.
We removed the cDNA amplification product from the thermal cycler. We have the SPRIselect reagent. Vortex until fully resuspended. Aspirate 60 microliters of SPRIselect reagent for each sample. After aspirating the desired volume of SPRIselect reagent, examine the pipette tips before dispensing to ensure that the correct volume is being transferred. Add the SPRIselect reagent to each sample in the tube strip and pipette mix 15 times with the pipette set to 150 microliters. Pipette mix thoroughly, as insufficient mixing of sample and SPRIselect reagent may lead to inconsistent results. Incubate the tube strips at room temperature for 5 minutes. Place the tube strips in a 10x magnetic separator HT in the high position until the solution clears. Carefully transfer the recommended amount of supernatant to a new tube strip without disturbing the pellet. Maintain at room temperature. Please refer to the protocol-specific user guide for details. Do not discard the transferred supernatant, as this is used to prepare the feature barcode library. If you are only preparing gene expression libraries, you may discard the transfer supernatant at this step. If there is additional supernatant remaining in the tubes with the pellet, remove and discard this remaining supernatant without disturbing the pellet. Do not discard the tube strips containing the SPRI pellet, as this contains cDNA that will be used to prepare the gene expression library.
Next, we will immediately proceed to pellet cleanup.
We have freshly prepared 80% ethanol. First, add 200 microliters of freshly prepared 80% ethanol to the pellet. Wait for 30 seconds. Carefully remove and discard the ethanol wash. We perform this 80% ethanol wash twice. After the second ethanol wash, centrifuge the tube strips briefly and return them to a 10x magnetic separator HT in the low position. Remove and discard any remaining ethanol. After removing the residual ethanol, let the samples air dry for 2 minutes. Do not exceed 2 minutes, as this will lead to decreased elution efficiency. Remove the tube strips from the 10x magnetic separator HT. Add 46 microliters of buffer EB. Pipette mix 15 times with the pipette set to 35 microliters and incubate at room temperature for 2 minutes. After 2 minutes, place the tube strips in a 10x magnetic separator HT in the high position until the solution clears. Transfer 45 microliters of sample to a new tube strip. For each sample, the amplified cDNA will be in 2 corresponding tubes. Transfer 15 microliters of cDNA from each of the 2 sample tubes to a third tube and pipette mix. Use this tube for downstream steps. Store remaining cDNA volume in tubes at 4 degrees Celsius for up to 72 hours, or at minus 20 degrees Celsius for up to 4 weeks for generating additional libraries.
Remember, if you are performing a feature barcode experiment, the transferred supernatant was set aside previously.
The next step for this transferred supernatant is to perform an additional SPRIselect and other cleanup steps. Please refer to the protocol-specific user guide for details.
We completed the SPRIselect cDNA cleanup. Please refer to the user guide for the remainder of the protocol steps.